everyone, and welcome to this episode of Talking RegMed. This episode we're talking ISSCR. It was the 15th annual meeting of the International Society for Stem Cell Research held in Boston, Massachusetts. Joining me and talking RegMed with me today is the commissioning editor for the award-winning journal Regenerative Medicine, Adam Price-Evans. Adam, thanks for joining me today.
Thanks for having me. Great to be here.
So let's kick off with the most talked about talk at this event, which is obviously Shinya Yamanaka's plenary on the first day. So Shinya Yamanaka was the discoverer of iPSCs, and his talk marked the 10th anniversary of his discovery. So Yamanaka discussed establishing a homologous HLA haplotype bank in association with the Japanese Red Cross. Now, he thinks that potentially one donor could cover up to 17% of the Japanese population. Adam, what do you think of his talk?
Yeah, it was great to hear from Professor Yamanaka and his work on the generation of safe iPSC lines. And they're looking at factors such as genomic stability after shipping. So this research is obviously very important, and it will hopefully bring forward the clinical use in the future. He also discussed an iPSC modelling example for fibrodysplasia ossificans progressiva, or FOP. And so using patient-derived iPSCs, they studied the cellular mechanisms of FOP, and they developed an in vivo model for the disease. After screening over 6,000 drugs, a novel drug candidate, rapamycin, was discovered, which inhibits BMP signaling and ossification in iPSC models for FOP. So this is a great example of how iPSCs can be used to model different diseases.
Now, at the other end of the week, in one of the last plenaries from ISSCR, was Masai Takahashi's talk on her iPSC therapy for wet-type age-related macular degeneration. Now, in the first clinical trial, she implanted a grown section of tissue from the RPE cells directly into the retina. You may have seen her interview on RegMedNet, which was ahead of her second clinical trial, which was utilising an RPE cell solution, again injected into the retina. I think what would have been really interesting in this talk is to hear her rationale on why the different mechanisms for the cell delivery. But she did mention that in the second trial, she was using Yamanaka-derived allogeneic cells as opposed to the autologous cells used in the first trial. So, Adam, what were some of your highlights from this year's ISSCR?
I managed to attend a number of concurrent sessions at the conference. For example, I went to a session chaired by one of our editorial board members, Nissim Benvenisti, and the session was on pluripotency and iPSC reprogramming. And one of the talks I found really interesting was from Catherine Plath from UCLA, and she was talking about how her team have been deciphering the mechanisms of the Yamanaka transcription factors.
Interesting.
And how they induce the events during reprogramming towards pluripotency.
Some of my highlights were, obviously we're doing the cell therapy regulation spotlight on RegMedNet. So it was really interesting to attend some of the ethics and regulation concurrent sessions. Again, sort of this is something that's been really popular at ISCT and World Advanced Therapies, and I think it really speaks to the fact of how far the industry is moving. Now, as always with these massive international meetings, the exhibit hall was packed. There were over 160 exhibitors from academic, industry, as well as startups. So Thermo Fisher had an update to their virtual reality experience that we saw at ISCT. This time they were covering different reprogramming methods for stem cells. So they covered lenti and retrovirus, episomal and Sendai virus. Biological Industries were also there. They were exhibiting high-performance culture mediums, and they took part in the Innovation Showcase. Ohad Kurniali from Advio Biotech talked about driving cell therapies to market. Millipore Sigma were there, exhibiting their technologies to advance innovative therapies, as well as the New York Stem Cell Foundation, the UK Stem Cell Bank, and Takara Bio. Out on the startup row, Cellink were exhibiting their new BioX bioprinter, which you may have seen announced on 3D MedNet. So, Adam, did you enjoy ISSCR? Any key takeaways?
As well as Takahashi's talk in the closing plenary, we also heard from Ole Lindvall from the University of Lund, and he was discussing sort of advances in the field since the first transplantation of human dopaminergic tissue in the brains of Parkinson's disease patients, which was done over 30 years ago. So that was great to hear from him. I
think for me it was really interesting to see sort of the commitment to open science and better science communication. There were loads of people live tweeting the meeting, sharing the results and the key presentations, which I think is really key for getting the public involved in regenerative medicine research and really invested in the future of these therapies. So that's it for this episode of Talking RegMed. I hope you enjoyed it, and if you've got any thoughts on ISSCR, let us know in the comments. If you're a RegMedNet member, look out for your exclusive Talking RegMed Extra video, which is also coming soon. So Adam, thanks very much for joining me.
Thanks for having me.
And if you'd like to appear on the next episode of Talking RegMed, let me know. Goodbye, and I hope you join us again soon.